Bismillah Rahman Rahim 5090 June 25 paper 4-2. This is O-level biology, and I'm going to explain some paper 4 skills to you all doing this exam. As you all know, this paper is for 1 hour and you should try to finish it in 45 minutes, because you can then check for your careless mistakes which you all make, and you're all very good at that. So please finish this paper in 45 minutes, and then last 15 minutes is for rechecking the paper. Now, please remember, when reading the question, you must highlight underline the words which are very important and which make sense, and you should read this at least twice. Catalase is an enzyme found in living cells. The enzyme catalyzes the breakdown of hydrogen peroxide into oxygen and water. So now hydrogen peroxide, and then it is broken down into oxygen and water. So make a mental note of this. Some plant cells are a source of catalase. If some material from a plant is crushed and added to water, a suspension of the contents of the plant cell can be obtained. Why is it a suspension? Why is it not a solution? Because the cell membrane is a phospholipid bilayer that doesn't dissolve. It suspends. Just like you make a banana milkshake, then the pieces of the banana are suspended in the milk. So it's not a solution. Glucose makes a solution because glucose dissolves in water. So please understand a suspension. If it's a green plant like leaves or something are crushed, then they've got chloroplasts in it. Chloroplasts don't dissolve in water. Mitochondria don't dissolve in water because they have a phospholipid bilayer, which is the membrane surrounding it. When hydrogen peroxide solution is added to the suspension, the oxygen produced is released as bubbles of gas. These bubbles collect to form a foam on top of the suspension. So this is the test tube, and we've got this here, and it forms a foam on top of it. I'm just drawing it in green. I don't mean the foam is green. It forms a foam on top of it. So the height of any foam produced can be measured using a ruler. Greater height indicates greater catalase activity. So if the height is more, say the height is now as big as this, as compared to this, so the height can be different. The height of the foam, please try to visualize it. Think you're doing this in the lab. I mean, I take my students to the lab. Even the grade 11 students go to the lab once a week, and they do a paper 3 so they can get ready for the paper 4. A student investigated the activity of catalase in the cells of 3 different species of plant by measuring the height of foam produced in each. They used 3 suspensions of cells, one each of celery, apple, and potato. So 3 different plants provided in separate beakers. The student followed these instructions. Use a clean stirring rod to stir the celery cell suspension in the beaker. Use a syringe to add 2 cm cube of celery cell suspension to a clean test tube. 2 cm cube, this is how we measure volume, to a test tube. Use a clean syringe to add 2 cm of hydrogen peroxide solution to this test tube. So you add another 2 cm cube of now hydrogen peroxide to this test tube. Immediately start timing. After 60 seconds, that is 1 minute, measure the height of any foam produced and record your measurements. So clean stirring rod, stir the celery suspension in the beaker. So you have this beaker full of celery suspension. So you stir it so that it all mixes up and the celery is not sort of settled at the bottom. And then we have some other liquid on the top. So it's a celery suspension. Then you use a syringe to add 2 cm of the celery to a clean test tube. This is when you added the celery here, the 2 cm celery suspension. And then you added 2 cm of the hydrogen peroxide. So this is the thing on top which I'm giving you a different color. And after 60 seconds, you measured now the height of the foam on the top. So this is the foam which was produced on the top. So the test tube would be, of course, much longer. So then you measure this foam, the height of this foam. Repeat these instructions using the apple cell suspension and then the potato cell suspension. Make sense? Now it says figure 1.1 shows the total contents of each of these three test tubes after 60 seconds. Now you can see this. This is a very nice picture of it. This is the potato, this is the apple, and this is the celery. Now the apple seems to have no foam, and the foam layer on this, and then we have the potato has. Um, Now you can see all they're all the same height. This one, and this one, because this is the same volume of the cell suspension and the hydrogen peroxide. Now this is the foam layer on top, so we'll need to measure this length. This is the foam layer on the top, and this we measured. And then the apple seems to have after 60 seconds. Then it says measure the height of any foam produced in each test tube and record these values in a table. Now the table rule is what? First is the independent variable which must go on the first column. Now when you make a table, always should be at least two or three columns depending on the information given to you. So drawn with ruled lines. So this is the first thing, is that it is drawn with ruled lines. And header, so what are you going to put in the heading? In the heading you're going to put here plant. Which plant did we use? So we are changing that we have decided to take celery and potato and whatever there is, celery, apple, and potato. So that is what is going to be on the first column. And then what are you going to measure? That is called the dependent variable. So height, please spell it correctly. Some of you even get this wrong. And then you put a slash, and then you write the units. I was doing this in class the other day, and students were writing height in millimeter. They were writing inward. I think that's a wrong thing. You put a slash actually. And there's only the Changat students who do this. And then you make further columns because there are three. So then you write here whatever you want to write. You want to write celery here, whichever you want to write first. I'm fine with that. I'm not saying there's any rule, but you see, if you look at this diagram, celery was first. So celery, apple, and potato. So celery, apple, and potato. Oh, there's no E in the end. So potato is fine with the written E. Then height in millimeters, you have to give that. And A, there was no foam. Or you can say 0 here. No foam, none, negative. So then you would say 23 and 13, that's in millimeters. Please do not write millimeter here. This is wrong. Do not write millimeter in each column. That is wrong, and you should not make this mistake. Now this is for 7 marks, but this table you see is going to be you're going to get the correct measurements was 3 marks, and the rest was 1 mark for the heading and 1 mark for the height, writing it in the millimeters, and all 3 plant types, and then correct measurements of the 3. Then it says part 2, use your results to place the 3 species of plant cells in order of catalase activity. So the more the foam, more the order of the catalase activity. So you can say celery had the greatest activity, then potato, and then apple, because apple seems to have no activity. So celery, potato, and apple got you 1 mark if you were very clear about, you see, more the foam, more the catalase activity, but that's what they told you in the question. So if you were reading the question properly, you would have figured this out. Suggest why the 3 plant tissues were crushed before adding the hydrogen peroxide solution. Why were they crushed? Because it said the plant cells contain catalase. So we need to break the cells to release the catalase. It's just like you've got a wrapper around your chocolate, so you've got to remove the wrapper to eat the chocolate. You can't eat the chocolate with the wrapper. I hope you're not that crazy, but then you're Gen Z, so I can't blame you. So suggest why the three plant tissues were crushed before adding to break open the cells, or you can say to release the enzyme or release catalase. Now please remember, when I underline something, like here I've underlined this, and when I underline it, it means this is an essential word that should be used. So if you said released cell contents, I'm not going to give you any mark. Release the catalase or release the enzyme, that must be written, and that's the words of the that's what the examiners are asked to check, that these words must have been used, should have been used. Then coming to the next part of the question, it says suggest why the plant suspension was stirred before adding the hydrogen peroxide. Why do you stir your tea before drinking it? Because you want to mix the sugar which might be at the bottom of it. So the ensure that the cell contents are evenly distributed. Evenly distributed measure ensure that the cell contents or the cells or the catalase is evenly distributed. That's why you're stirring it. And or you can say maximum surface area for enzyme activity. Enzyme activity the same for each species. Please remember that always look at the marks. If there are 2 marks, then they must give me 2 points. For just something 1 point, you can't be giving you 2 marks. There's no for some irrelevant point you expect me to give you the 2 marks. So when I'm checking your papers, I need to see that you've given me 2 separate points. So ensure that the cell contents are evenly distributed, and to maximize, maximum surface area for enzyme activity for each species, because you see, otherwise the cell suspension might settle down and there just be water on the top. Now, part 3 says state one variable that was controlled in this investigation. What is the variable that was controlled in this investigation? The volume of the hydrogen peroxide and the volume of the cell suspension or the plant tissue or the plant suspension. 2 ml of that and 2 ml of the hydrogen peroxide. So there were 5 variables that were controlled in this. The volume of the plant tissue was controlled. The volume of hydrogen peroxide was the same in each. The time, after a certain time, how much, that was the same, 60 seconds, and the diameter of the test tube. So all these, any one of these was, you could have got your 1 mark for it. It's only for 1 mark. So any one of these, but you should know. I mean, I could have asked this, given you say 5 marks and said, okay, which variables have been controlled? So you should know that volume of the plant tissue was the same, volume of H2O2, the time after 60 seconds, the foam was measured, and then the diameter of the test tubes were all the same. Then suggest 2 reasons why repeating the investigation would give the student more confidence in the results. Now, we explained this in many papers, and that is that, identify anomalies and to make the results more reliable. We just have to write these 2 things, and that is what will get you your 2 marks. So now these are difficult words, anomalies and outliers. So please get comfortable using these words and spelling these words. Outliers means, yeah, sort of an error. Like the trend is, say, the results were 10 and then 11 and then 12, and then one result is 25, and then again it is 12. So, I mean, the trend is 10, 11, 12, and 12, and this is now an anomalous reading or an outlier. Outlier, not liar. The liar that we say you're a liar, that's something, this is a different spelling. So then the anomalies is that it doesn't fit a trend and it is an anomalous reading. We're not going to ask you why. It could be an experimental error, could be some other error or something. You'll do that in A levels, but I know most of you will not do A level biology. Now we come to the part C of the question. Another student used the same procedure using a celery cell suspension and hydrogen peroxide. Instead of using a test tube, they used a measuring cylinder. They measured the total volume of the contents of the measuring cylinder every minute for 5 minutes. These measurements are shown in table 1.1. The student did not record the measurement at 4 minutes. So they measured the total volume of the contents of the measure every minute for 5 minutes. but then did not record them at 4 minutes. So there is a 4 1 is missing here. So this 1 0 1 2 3 and then 5 minutes. Interesting. Then it says construct a graph of the data in table 1.1 on the grid below. Join your plotted points with ruled straight lines. Now the first thing to do is that you label the axis. This is the first law. Label axis. So you go back here and you see what is written here. It says time and minutes. This goes on the x-axis. This is always the rule. This is the independent variable because you have decided to measure them every minute or for 5 minutes. So this will go on the, so this is the first column. This is for you all to remember because you all have very short memories. And this is always the independent variable which you and me are changing. Variable. Again, they're going to ask you this in another paper. So the mnemonic is FIX. Well, you all need to be fixed also. So time in minutes will go on the x-axis, and this total volume of contents and the whole of it, nothing should be abbreviation. All of this goes on the y-axis. This is the rule that we follow. So please first of all write that before you do anything of the graph, because if you do this you get 1 mark for doing nothing, for just writing this down from the question paper onto the graph paper. So just writing these 2 points will get you 1 mark, and if you don't write it, well, that's a sad thing because you lose that mark. So time on x-axis and both axes fully labeled gets you 1 mark. Then you decide the scale. How do you decide the scale? Let's look at that and what is given on the so we have the smallest number is 0 and then we have 5. So 0 to 5 on the x-axis. So let's now look how are we going to do this. So it's very easy because there is 0 here, and then for every 2 centimeters, so this will be 1, this will be 2, this will be 3, this will be 4, and this will be 5. So that's very easy because we have to do it from 0 to 5. So we do that, then we go back and we say now what is the figures. It's the smallest figure is 4, and then we have 20.5. So then we have to adjust this on this side. So then we do it, then we write the 0 here for the y-axis as well, and we have to get it to 25, 5, 10. So this we have 5, 10, 15, 20, and 25. So we've used the whole grid, and that is the most important thing that we do, is that we use the whole grid. So we've decided and we've got to label it every 2 centimeter. So a good use of the grid. Then you start correcting it, you start plotting the points, and then you join the points with a rule line, and there should be no extrapolation. Extrapolation means do not extend it from the points that are given in the paper. So then you plot the points. How do you plot the points? Either you put a dot and a circle, or you put a cross. I usually like a dot and a circle, and please, it should not be a blob and a circle. This is the dot and the circle. This is a blob. This shouldn't be. This is wrong. If you put a cross, then only put a cross. Then I don't have to circle the cross. And then you join these with a ruler, a point to point with a ruler, very point going from one point to the other. Very exact, very properly. Now please remember that the line must go to the point. Like this is the point and this is the point, so it should be joining the points. Some of you made a point. I remember checking this yesterday, and somebody had made the points like this. Now this is going below the point. Now this you will lose 1 mark because joining the points is a very basic skill, and if you don't have that at O levels, then I think we will have to decide something else for your life. Maybe give us Root Rishta ka ad. So join the points with a ruler, and don't make this mistake, because the 4 marks, these are I call them मुफ्त के 4 marks हैं, ये आपको वैसे ही मिल जाएंगे. So you really have not worked. There's no bio in it. There's no bio that you knew. You have to just do this graph, label the axis, decide the scale, and then plot the points and join the points. That's it. It's sad if you lose any mark on this. It's Very sad, very sad. Then next part of the question says use a graph to estimate the total volume of the contents of the measuring cylinder at 4 minutes. Show your working on the graph, total volume at 4 minutes, and there's 3 marks for that. Now, yesterday I was doing this in class and the student didn't read this. And they said, oh, miss, I didn't read this. And so if you read and yet you do not read, I'm sorry, then we need to reassess your brain functions and maybe you need an MRI or something to find out if your brain is functioning or not. Now you need to draw a line at 4 minutes. Maybe my line is not very good. I know I didn't use a ruler. So at 4 minutes, it says show your working on the graph. So I have to see this line. This line gets you 1 mark. And then the fact that 20 gets you another mark because I'm reading off it's 20. So what is the third mark for? Which 50% of the students lost was you didn't write the units. cm cube. You have to write that. So 1 mark for this line on the graph, 1 mark for this, and 1 mark for the unit. Because you are inevitably going to forget the unit. So 3 marks is like that. Then it says part 3, describe and explain the shape of your graph. The shape of the graph was what? Let's look at the shape of the graph. Describe and explain the shape of the graph. So volume increases with time. Yeah, fine. Volume increases with time. That's what you can see here, that the volume is increasing with time. And then what happens? It plateaus. Or you can say the rate becomes constant, or the rate reduces, or the rate becomes constant. Total volume of, no, the rate doesn't become constant. The total volume becomes constant. So the rate plateaus, or you can say volume increasing in a given period of time, and then of course the volume is not increasing. So the total volume is remaining the same. So it's constant. So what has happened is all the hydrogen peroxide has been used up by it. So why has this is because you see on the y-axis you have the total volume of contents. Now why is the total volume remaining the same? Because the enzyme has used up all the hydrogen peroxide, and that is why now no more foam is being made or no more of the substrate is available. So how can we get the foam now? So hydrogen has become the limiting factor here. So this will get you your 3 marks. Volume increases with time and then it plateaus, or you can say the rate reduces. I would say volume of content remains the same. Hydrogen peroxide used up or broken down. Now coming to question number 2, figure 2.1 is a photo micrograph of a section of a celery plant. In the space below, make a large drawing of the plant section as it appears in the photo micrograph. Now please remember this was for 5 marks. I mean, I've drawn a smaller diagram, but it should have been a clear, clean, continuous lines drawn with a sharp pencil and no shading. Then it should be at least 90 millimeter minimum width from A to B, and concave bottom edge. So this is the concave bottom outer edge and the scalloped upper edge. Then detail of ridges on the upper edge, double line on the scalloped upper edge, and delimited bumps, colon chyma, and then the vascular bundles drawn. So this would get you your, please draw a big diagram, draw what you see, and please give me all the details which are on the diagram. Then it says draw a straight line to join A and B on figure 2.1. This is the length of the plant section in the photo micrograph. Measure and record this length. So you draw this line on the diagram and you measure this length. This was about 59 millimeter. So you write this here, 59 millimeter. Millimeters of course given here, so I don't have to write this again. So 59 millimeter was the length, and that got you a mark, but they've allowed you anything between 59 and 61. So you could have made that. And then it says on your drawing, draw a straight line. On your drawing, draw a straight line in the same position as line AB you have drawn on the photo micrograph. Measure and record the length of this line. So then you measure this one on your diagram. So you need to then measure this part now. I'm going to use another color. So you're going to measure this part now on your drawing. You have to measure this part. So this would be the part A, B. You don't have to write this. Then it says on your drawing, draw a line. So I mean, I haven't measured it. I don't have a ruler. So I'm assuming that it's 100 millimeter. So 100 over 59, whatever your measurements for. I'm not saying it's going to be 100. Your diagram could be something else. Could be 110. Could be less. Could be more. So your drawing will be dependent on that. So main you have to do is divide the drawing by the actual, and that gives you the magnification. So it's 1.69 because it says two decimal places. Please understand what is two decimal places. Two, after the decimal there should be two numbers. Last question is the planning question. So plan an investigation to find out the effect of varying light intensity on the increase in height of mustard plant seedlings provided in petri dishes as shown in figure 3.1. So moist filter paper, petri dish and seedling. Now, what is the thing you're going to change is the light intensity. So at least three different light intensities used. Then how would you change the light intensities? Move the lamp, or you can say you can add different bulbs, or you can say have a dimmer switch. Then light source from above only. Then the same time between measurements. Then control the variable, same volume of water or nutrient. So we've got about 8 points, and you could have given me any six to get these 6 out of 6. So three different light intensity were used. Use different bulbs, or use a dimmer switch, or you can move the lamp. Then light source from above only. Then rest of the room should be in darkness, because you see otherwise there are other lights in the room as well. So you can't say it's only because of the lamp. Leave for 2 to 3 days, because you can't see them growing in a few hours. You'll have to leave them for even more time. And then you should have measured the length in the beginning and then the length after the 3 days. So same volume of water and temperature. This is the other variables that you control. Control the other variables. That's very important that you know independent variable, dependent variable, and the control variables. Then you should know the starting and the final length. Then the same volume of water or the same temperature should be maintained. And if you are adding any nutrients, then that should be the same in all the situations. Then you calculate the increase in height per light intensity, because you've used three different light intensities. So different light intensities would mean different patterns of growth, or different more or less growth, more or less. So you could then prove the fact and say, yes, when we had more light intensity, so this happened, or less light intensity. But we're not asking you for conclusions. We're just telling you, we're just asking you to give us how you would conduct this experiment. So that completes this paper, and thank you very much and all the best.